Namaste students, खुश रहो बेटियों. I Iam Dr Chandrakala, Assistant Professor of Vidyapeeth, University, Jaipur. In today's session, we are going to discuss about the pollen culture. The microspores or the immature pollen grain can be used as an explant to get a haploid plant directly. For pollen or a microscope or a microscopic like the spores, microspores culture, the flower buds are collected. The surface sterilization and the anther lobes are dissected out from the flowering bud as before, and then the anther lobes are squeezed with the help of a scalpel within a tube, and then a small beaker to collect the microspores or a pollen in a nutrient medium. So here the anther tissue debris is removed by filtering the suspension through the nylon seeds or with a diameter slightly larger than the pollen size. Okay, so allowing the microspores only to pass through. So the microspore suspension washed and concentrated to a plating density, and the microspore obtained then mixed with the appropriate culture medium at a density of 10 to the power 3 to 10 to the power 4 microspore per ml and plated in a small petri plate. To ensure an aeration, the layer of the liquid in a dish should be thin as possible and sealed with a paraffin to avoid the dehydration. So the response of the pollen will be divided into in the form of embryo or a gamete. So when directly or indirectly will form a haploid plant. Okay. So So here in the slide screen, you can see the plant like the flower, which is like surface sterilization of the flower was done first, and with the split of the flower bud taken out of the stem, and here the discarding the filaments. We have just removing the first the plant flower pollen with the filaments, and here after the filament is removed for the pollen. So here gently the crushing of the anther spine with the help of a glass rod, and then with filtering it with a sieve of 10 micro and 40 micro depending on the size of the pollen and into a MS broth. Okay. So here the pollen suspension is collected, and that was reduced to 800 to 500 to 800 rpm for 50 minutes. Okay. So the sedimentation will take place of the pollen grain. Then the resuspension is fresh medium in a broth and centrifuged again. So here the pellet of the pollen is formed, and from that the pollen is obtained, and that is incubated in a plate of 26 to 24 to 26 degrees centigrade for 3 to 4 weeks in a petri plate having the MS medium. Okay. So from where it can develop into embryoids and then to regenerate into a complete haploid plant. Okay. So the advantages of the pollen culture is the explant and the microspores or a pollen grain of all the haploids are of all haploids. Thus, a sequence of the embryogenesis can be obtained starting from a single cell. The microspores are ideal for take-up of and transformation of the mutagenesis studies. High yield of the plant or anther could be obtained. Okay. Thank you. This session is powered by 2.0 Vidyapeeth Women's University, Jaipur. If you have any queries,